Today, we're diving into one of the most incredible processes in all of biology, DNA replication. It's this microscopic marvel that lets the entire instruction manual for a living thing be copied almost perfectly, billions and billions of times. It's happening inside you right now.
So take a look at this molecule. It's in every single one of your cells. And for your body to work, each new cell needs a perfect copy. Before any cell can divide, whether it's for growing, healing a cut, or just replacing old cells, it has to duplicate its entire library of genetic information. It's a monumental task. All right, let's get into it. Section 1, Life's Ultimate Blueprint. So what exactly is this blueprint and why is copying it so absolutely critical? Well, DNA is basically the cell's master instruction manual. It's a double helix, which you can just picture as a twisted ladder. The rungs of that ladder are made of 4 chemical bases: adenine, or A, which always pairs up with thymine, or T, and cytosine, C, which always pairs up with guanine, G. This super strict A with T and C with G rule, that's the secret sauce. It's what makes copying it so incredibly accurate. Now, the process itself has a pretty cool name: semi-conservative replication. And what does that mean? Well, it's actually simple. When DNA gets copied, you don't end up with one old molecule and one brand new one. Instead, each of the 2 new DNA molecules is a hybrid. It's made of one of the original strands and one freshly made strand. It's a beautiful way to preserve the original template perfectly. So, Section 2, meet the replication crew. You might be thinking this is a simple job, but oh no. This is a complex operation, and it takes a whole team of highly specialized enzymes to pull it off. Think of it like a molecular construction crew, where every member has a very specific job to do.
And here are the stars of the show. First up, you've got helicase. That's our unzipper. Its whole job is to pry apart the 2 DNA strands. Then comes primase, the initiator, who lays down a little starting block so the builder knows where to get to work. The master builder itself is DNA polymerase. And finally, you have ligase, the gluer, who comes in at the end to seal everything up and make it perfect. Okay, so just like any construction project, the first thing you have to do is prep the site. Before you can build anything, you've got to open up that blueprint. And in the world of the cell, that means unzipping the DNA double helix. This Y-shaped area you see here, that's our job site. It's called the replication fork, and it is the exact spot where all the action happens, where the DNA is actively being unwound and copied. So let's break down how this site gets prepped. First, helicase gets in there and starts breaking the bonds holding the 2 strands together, zip. Then, these little things called single-strand binding proteins, or SSBs, clamp onto the separated strands to keep them from snapping right back together. And all the while, another enzyme called topoisomerase is working just ahead of the fork, relieving the tension that builds up from all that twisting and unwinding. It's a real team effort.
Now the site is prepped, the crew is ready, and construction can finally begin. But this is where things get really clever, and frankly, a little complicated. It's all because our master builder has to follow one very, very strict rule. And because of that rule, we run into a fascinating problem. How in the world do you build in 2 opposite directions at the very same time? Here it is. This is the crucial rule. DNA polymerase is basically a machine that only works on a one-way street. It can only add new DNA bases to one specific end of the growing strand, what's called the 3' end. So it can only build in a 5' to 3' direction. This one simple rule makes everything so much more interesting. See, the 2 original DNA strands are anti-parallel, meaning they run in opposite directions. Because of our builder's one-way rule, the 2 new strands have to be built completely differently. One of them, called the leading strand, is a piece of cake. It's built in one continuous piece, like driving down an open highway. But the other one, the lagging strand, is more like trying to drive through heavy stop-and-go traffic. So, what's the solution for this tricky lagging strand? It's ingenious, really. Since the polymerase has to build away from the direction the fork is opening, it just does it in short, little discontinuous bursts. And these little segments have a name. They're called Okazaki fragments. So here's the play-by-play. For each new fragment, primase lays down a temporary starting block. Then, DNA polymerase jumps on and builds a short stretch of DNA, an Okazaki fragment, until it bumps into the primer from the previous fragment. This just happens over and over again, as the fork keeps opening. Finally, our trusty gluer, ligase, comes in and stitches all these fragments together into one solid, continuous strand. So, just to lock that in, Okazaki fragments are the brilliant solution to that one-way building problem.
They are the short, separate pieces of DNA that allow the lagging strand to get built, even though its template is running in what you might call the wrong direction for our builder. And get this, there's even a cleanup crew. After the main high-speed builder, DNA polymerase III, zips along making most of the new DNA, another specialist, DNA polymerase I, comes through. It's the repair and cleanup expert. It removes those temporary RNA starting blocks and fills in the gaps with the proper DNA, making sure the final product is perfect.
So, after all that unzipping, priming, building, cleaning, and gluing, what are we left with? It's time for the grand finale of this incredible molecular performance.
And here's the final result. 2 absolutely identical, complete DNA double helices. Each one is a perfect semi-conservative copy. Remember, that means 1 original strand from the parent and 1 brand new strand. The blueprint has been duplicated successfully. Mission accomplished. But you know what's truly mind-blowing? The accuracy. DNA polymerase isn't just a builder, it's also a meticulous inspector. It has a built-in proofreading function, so it's constantly checking its own work and fixing almost any mistake on the spot. And this is so critical, because even a single uncorrected error could lead to mutations. This proofreading is the cell's first line of defense, making sure the code is passed on with incredible fidelity. And this isn't just some abstract thing happening in a textbook. Understanding this process is at the very core of modern science. It's what powers biotech, like the PCR test. That's basically just DNA replication in a test tube. It's also vital for medicine. A lot of cancer drugs and antibiotics actually work by messing up this exact process in the cells we want to get rid of. So if you remember just 3 things from all this, let it be these. First, replication is semi-conservative. 1 old strand, 1 new strand. Second, it takes a whole coordinated team of enzyme specialists to pull it off. And third, the whole thing is astonishingly accurate thanks to that built-in proofreading function. And that leaves us with one last thought. This incredible process is happening billions of times a day, right now, inside your body. But with that many copies being made, near perfect isn't always perfect. So, what happens when one of those incredibly rare mistakes does manage to slip through the cracks? Well, that single tiny error is where the whole story of mutation, genetics, and even evolution begins.